Hello, everyone. My name is Yunxi Liu. I'm a PhD student at Rice University Computer Science Department. I'm a member of the Treangen Lab. Today, I will be talking about rescuing low-frequency variants within intra-host viral populations directly from Oxford Nanopore sequencing data, also known as the ONT data. But first of all, let's go over the learning objectives of this presentation. Today, I will introduce variant detection of viral genomes. I'll explain within host consensus level and low-frequency mutations, and I'll be identifying the current challenge of variant detection in Oxford Nanopore sequencing data. And at last, I'll introduce a computational method to distinguish between the ONT sequencing errors and the true biological variants. First, uh, let's look about the variants. There are two types of variants we're going to mainly discuss in our presentation. The first one is called single nucleotide variants, also known as SNVs. This occurs when a single nucleotide, for example, A, T, C, or Gs, is altered in the genomic sequence. Also, there's another type of variant called indels, which are insertions and deletions of nucleotides into the genomic sequence that are less than some length. In this case, the indels we're talking about has the length of 1 to 50 base pairs. So the study of variants have very broad application in bioinformatics, such as subcolonial evolution in cancer, bacterial subpopulations, and also it's one of the key studies for intra-host variants in RNA virus, such as SARS-CoV-2. This is because RNA virus, they usually have high mutation rates, they have high replication rates, and those variants, they affect the toxicity, transmissibility, and also what's more important, they affect drug treatment strategies and genetic monitoring of the live vaccines. So on the right, you can see a figure of the phylogenegy of the SARS-CoV-2 virus and how people determine whether a different strain of SARS-CoV-2 belongs to Alpha or Delta or Omicron. Those are all determined by the SNVs and indels found in each of the strains. So why do we care about low-frequency variant? For low-frequency variant, what does it? It means that the allele frequency, which is the proportion of the variants, occupies less than 50% of the total population. The reason we care about low-frequency variant is that they change over time. In this figure, I'm showing an observation of the viral population of the flu virus in pig model. As you can see from the figure, there are some of the variants, they start with very low allele frequency below 0.1, and in a 4-day period, the allele frequency of those variants increases. At the end of day 5, some of the variants had reached the allele frequency of 0.4 or 0.5. And this is not only happening in the individual individual host. In an active outbreak setting like the COVID pandemic, the low-frequency variants can become dominant over time, reducing the sensibility of the detection method. Here I'm providing an example of the variants. One of the well-known variants in the early pandemic is called spike protein D614G. This variant is caused by an A2G nucleotide mutation at location 2343 in the Wuhan reference strain. If you look at the global transition plot on the top right, you can see that at the very early stage of the pandemic, around mid-February 2020, the D614 kind of dominates the entire proportion of the different SARS-CoV-2 strain at about 100%. And just within a month, The G614 actually gained its popularity and its frequency went over 50% and become the major variant. And as time goes by, the G614 eventually kind of have a frequency of 100% totally wipe out the D614 variant. Studies have shown that patients infected with G614 shared more viral nucleic acid compared to those with D614. And the new variant G614 bearing virus shows significantly higher magnitude of infection compared to the old D614. So let's now talk about variant calling, like how those variants are being detected and what is variant calling about. So variant calling is a central task of genomic analysis. It actually answers the question of how to distinguish between the true biological variation or mutations and the experimental errors. They often require sophisticated statistic, computational, or heuristic techniques. So the question finally comes down to if a sequencing technology has an error rate of 1%, can we call variants with the allele frequency of 1%? Or even more, what if the sequencing error is around 10%? Can we actually identify variants with allele frequency below 10%? So this figure shows the main challenge of variant calling. So with the read aligned to the reference genome, where the reference genome is shown on the bottom of the figure, we can see there are some bases that are different from the reference. And the variant calling process answers the question that is this base a true variant or true mutation, or is it a sequencing error? So keep in mind that the sequencing error rates varies significantly between technologies, substitution types, and many other factors. Uh, so if we like zoom out a little bit, looking at the what the actual alignment looks like, this is an alignment generated from the Illumina parent sequencing dataset submitted early in the pandemic by the Wuhan University, and the reads, which are represented in those red and blue segments, are aligned to the NCBI reference sequence for the SARS-CoV-2 virus. And if we take a closer look at the alignment, where the reference genome is still the bottom row, we can see supermajority of the bases are identical to the reference genome. That's why we say they're aligned. And if we mask those bases that are identical to the reference sequence, we're left with those substitutions and also deletions and insertions, where deletion is represented by a black line, and the insertions are labeled with how many bases are being inserted. So the variant calling tools actually trying to figure out if we have a bunch of different bases in a column, is that indicates that's a true intra-host mutations, or is it just the noise caused by the sequencing error? So next, I'm going to talk about why variant calling using ONT data is difficult. So before the third gen sequencing came out, the variant calling task is often done with the next gen sequencing platform represented by Illumina. Those second gen sequencing, they produced short reads with very high accuracy. So the base call accuracy for Illumina can go up to more than 99.9%. But now we have the third gen long read sequencing technology, which they produce much longer reads and with low cost and portability. But there's a trade-off that the error rate in those ONT data are very high compared to the Illumina sequencer. The error rate can go up to 5% to 15% in the data, so that makes the variant detection extremely challenging for those ONT data, especially if you want to detect intra-host minor variants, which are low frequency variants. And also, there are systematic errors in the ONT data that might be supported by the majority of the reads. So as a result, the existing method for variant calling for ONT data, they are all haplotype variant callers, which means they do not target low frequency variants. In fact, there are some current polishing method or error correction method for the ONT data, such as Recombinatica pipeline, Nanopolish pipeline, and those tools use graph-based or machine learning approaches to try to correct the errors in the ONT data. But what they want to do, their focus is for genome assembly instead of variant calling for their downstream analysis. So during their process where they're doing the error correction, they actually eliminate the true signal for the low frequency variant. So you cannot just run error correction method, then do low frequency variant calling on the corrected data. Luckily for us, there are some patterns hidden inside of the sequencing error in the ONT data. First, the error rate in the Nanopore sequencing data is context dependent. And also the indel errors in the ONT data, they often occurs at the low complexity region. What those means, I'll explain in the later slides. So we come up with the hypothesis that the allele frequency for the true SNVs is subject to change across samples, while those sequencing errors are independent of samples, thus are highly stable or highly similar. It is possible to recover intra-host variants for ONT data by exploiting the tendency of true variants to change in allele frequency across multiple samples. Here, I'm showing a set of samples that are collected with a COVID positive patient and one individual, and those samples are collected at different time points. Then the sample got sequenced on the on the Nanopore sequencer, and what we're looking at are the non-reference space, which is the sequencing errors at different precision of the genome. If we take a closer look, we can see that for those different samples at each of the position, they actually have very similar error rate. Or say, if you look at the trend of the errors, they're actually multiple samples, they share the same pattern. And if we look at the indels, which are insertion and deletions, here I'm showing a histogram of 20 most frequent fimers that deletion occurs. If you look at the labels, you can find those indel errors, they're actually occurring most likely in the homopolymer regions. Here, homopolymer means the same nucleotide, for example, T, getting repeated over and over again. Whereas the first label, we have 4 T's clustered together. And what about the true biological mutation? Here I'm using a very simplified toy example to show that why the true biological mutation have their allele frequency changes over time. For example, there are 2 strains of SARS-CoV-2 virus in a host, and strain A has 9 copies on day 1, and strain T, which containing an A to T mutations on some location, has only 1 copy. And on day 1, if we calculate the allele frequency for this mutation, that's going to be 1 over 10, which equals 0.1. And those two strains, because of the mutation, they have different replication rate, and the strain T has much higher replication rate. So on day 2, strain A doubles, which gives that 18 copies, where strain T already got 10 copies. So if we calculate the allele frequency for this mutation, that is going to be 10 over 28 equals 0.36. And as you can see, the allele frequency already increases. And on day 3, strain A got 36 copies, while strain T already is at 100 copies. So now the allele frequency for this A to T mutation is going to be 100 over 136, which is equal to 0.74. That makes this mutation a consensus level mutation, which means the allele frequency is above 50%. So in this example, we see how a true biological mutation have their allele frequency change over time. So with those background knowledge and a hypothesis in mind, we build tools called Variable that is able to let people to perform variant calling with ONT data directly. So you have the patient, you collect the samples from the patient, and the samples are sent to lab for lab preparation, and you put the samples on the sequencer. The Nanopore sequencer gives you a bunch of reads. You align the reads to the reference genome. Then you try to do the calculation column by column to identify the variants. This is the kind of a standard approach using a standard variant calling pipeline. But here, since we're using Oxford Nanopore data, which has a much higher error rate, the variant calling result will contain a lot of the false positive calls. That's where Variable kicks in. Variable, it actually uses computational method with its two filters. One is called Crosssample allele frequency variation filter, and the other is Intel low complexity filter to kind of identify which within the variant calling result, which ones are the false positive calls and which ones are the valid calls. At the end, it outputs a set of variants that is highly accurate. So let's take a deep dive into how those two filters works. First is cross-sample allele frequency variation filter. So Variable actually examines each one of the VCF file, which is a file format that containing the information of the variant calls, identifies the variants that are shared between samples, and records their allele frequency. Then it eliminates false variant calls if the cause has a maximum allele frequency less than 0.65 and maximum allele frequency variation less than 0.05 across all samples in which the variant existed. Here is a very simple toy example for that. We have 4 samples collected from an individual on 4 different time points, and we have 3 variants in this sample. For variant A, we can see it has a pretty high allele frequency near 1, and the allele frequency does not vary much. In this case, the variant A is going to be classified as a valid variant because those kind of variants are actually associated with a mutation at the frequency of 1. It is just because the nanopore sequencing error, so in the output, it shows that the variant has the allele frequency less than 1. And also we have variant B, which has a lower allele frequency, but the allele frequency varies between time points. This indicates that variant B is a true biological mutation. And also we have variant C, which has pretty low allele frequency, and the allele frequency does not change much, which indicates that the variant C is a false positive cause uh, made by the variant caller because of the sequencing error. So variable actually detects those kind of variants and mark them as invalid and eliminate them during the process. Also, we have the low entropy filtering designed for the indels. It is designed to eliminate nanopore homopolymer errors that primarily occurs in regions with short repeats. Here we have another example. So how the algorithm works is that assume a deletion is called at some position on the reference genome with some length. The filter actually checks the product of the Shannon entropy, which is indicating how complex this sequence are for two substrings, the substring A and substring B. So sequence A is actually a small segment of sequence before the indel happens, and sequence B is the sequence that the on the other side of where the indel happens. So if the value of the product of those Shannon entropys of those two sequence A and sequence B are less than some usually defined threshold, by default is 1, that means either sequence A or sequence B or both, one of them are actually contains low complexity sequences. Then the variant is classified as false cause and it is eliminated. So we test our tools variable on two COVID datasets. One of them is a longitudinal dataset, and it contains time series data collected from a single patient periodically. There are 23 samples in total collected over 101-day periods. All samples when sequencing with the ONT platform, and 20 of them when short-read deep sequencing using Illumina platform. There's a reason we want to include the dataset containing both ONT and Illumina data is that with the highly accurate Illumina data, we're able to generate a set of high confident variant calls that are used as a ground truth to benchmark our method. There's also a cross-patient dataset containing 145 COVID positive samples collected from different patients, and also the dataset is sequenced with both Illumina and ONT. And here we're looking at the result. This figure shows the precision, the recall, and F scores of the variant calling result with the ONT data on a standard variant calling pipeline and with the variable attached to it. And also we're benchmark variable against one of the state-of-the-art variant caller for ONT data, which is that's called CLAIR3. Here I have to mention that CLAIR3 is not designed for the low frequency variant calling, but it is the best variant caller for ONT that we can find. So from the result, we can see variable did significantly better than CLAIR3 in terms of precision on both of the data set, and variable has significantly higher F score in the time series data set. And if we look at the Venn diagram, this shows similar result where the big red circle is the ground truth, and we can see variable actually produce much less false positive calls compared to CLAIR3. And if we look at the false positive rate at different allele frequencies and the accumulated count of the false calls made by variable and CLAIR3 for the time series data set, we can find that variable has much lower false positive rates for variants that have allele frequency below 0.65 compared to CLAIR3. And the peak false positive rate for variable occurs at the allele frequency between 0.2 and 0.25, which we believe that is associated with the sequencing error rate. Also, it has a close to uniform distribution, where CLAIR3 they have their false positive call mainly cluster at low frequency range, which is below zero point 0.5. We also did a sanity check for variable using the synthetic data. So the synthetic data set is constructed with a viral negative nasopharyngeal swab spiked with plasmas containing the synthetic S and N gene of the SARS-CoV-2 reference genome. There are 112 samples sequenced with the ONT platform, and we use that as a negative control to evaluate variable's false positive rate. Since it is synthetic data, it contains no true biological variants or biological mutations. Therefore, all variant calls made are classified as technical artifacts or error. If using ONT data directly on the variant calling pipeline, as we can see from the figure here, left figure, the blue bars before applying variable, we can see there are so many false positive calls. And after applying variable, almost all the variant calls are being eliminated. If you increase the minimum coverage that's supporting the location to 50, our false positive rates drop to below 0.01. And if you keep increasing the minimum coverage to 200x, there's no false positive calls on the synthetic data. And on the right figure, we're showing how each of the filters performs. As we can see from here, most of the arrows in the synthetic data set, they are associated with the indels, and it is removed by the low entropy filter, showing green. And also, the rest of the SNV arrows are removed by the AF variation filter, showing orange. We also expand the usage of variable on non-COVID dataset. On the top figure, you see it is Ebola virus dataset. On the bottom figure, that is the norovirus dataset. And variant calls before the filtering are shown in blue, and variant calls after applying variable are marked in red. And this shows that the variable is capable of removing noise while still identifying a large number of low-frequency intra-host variants in the sample. Last, I want to talk about some limitations for variable on the cross-patient studies. The main cause of this limitation is that the same variants must be observed at least twice in the sample to activate the allele frequency variation filtering, and the sample collected from different patients are less likely to have them. Based on a single simulation, assuming the variants occur randomly, we see we need like 10,000 samples to recover the supermajority of the variants, which means the variants are shared by at least 2 samples. So as a result, we see a drop in the average precision in the cross-patient dataset compared to time series dataset. For the same reason, performance of variable may be affected if fewer samples are available even for the longitudinal data. But luckily for us, both scenarios could be improved by leveraging a centralized data depository of low-frequency INCVs for SARS-CoV-2. And in conclusion, we have shown that by comparing the allele frequency of the same variants across multiple samples, variable is able to distinguish true biological mutations from ONT sequencing errors, even at a low frequency. And it is the first method explicitly designed for such task, and this represents both an important advance for the field and will facilitate the tracking of intra-host variant in patient samples for COVID and other diseases. Variable is open source and available for download at the following link, or you can just scan the QR code. And here, let's review our learning objectives. In this presentation, we introduced the variant detection of viral genomes, explain the intra-host consensus level and low-frequency mutations, identify the current challenge of variant detection using ONT data, and also introduce the computational method to distinguish between the ONT sequencing error and the true biological variants. At last, I want to thank Treangen Lab, my mentor, Dr. Todd Treangen, Joshua Canali, Bryce Killer, Dr. Michael Nude. I also want to thank Sadrzak Lab, Dr. Fritz Sadrzak, and Dr. Madhab Mohammed. I also want to thank the contributing authors of the Illumina and ONT sequencing data, which was instrumental for highlighting the benefits of variable or ONT data. At the last, I want to thank all the founding and supporting agency that behind this project. That's all. Thank you for listening.